and thank you for the introduction and thank you for the invitation to speak about this work. So we're looking at humans, another human talk, and we're trying to answer questions such as how does the quality of frequency representation affect our ability to perform complicated auditory skills? We're very interested in things that only humans do, like play musical instruments and have long, complex conversations. But specifically in this talk, I'll be focusing on something called hearing and noise perception. And that's because it has a lot of clinical relevance. So it affects social, educational, and vocational interactions and quality of life, particularly amongst older adults, which is a focus because many societies, of course, are having aging populations at the moment. It also affects school children and their ability to understand in noisy environments. It can occur in the absence of peripheral hearing problems, which means that just checking someone's audiogram is not enough to understand if they have a hearing and noise issue. So I don't need to tell anyone in this room that the auditory system is for encoding vibrations in the external environment that for us are transmitted through air. And in the human brain, some of the main areas that are of interest are cochlear nucleus, inferior colliculus, medial geniculate body, and auditory cortex when we're talking about frequency representation. But that's not all. This becomes relevant to just see the anatomy because in humans, unless you're putting in intracranial electrodes, which you can't do in healthy humans, of course, these nuclei are all located some centimeters away from each other, or at least 1 centimeter away from each other. And what that means is we can actually try to separate them out using some high-density EEG recordings or magnetoencephalography recordings. From the auditory cortex, of course, we have streams of information that pass this frequency information to other places in the brain, and there they are used for all kinds of other interactions and integration with other sensory modalities and such. When we're thinking about hearing and noise perception, that's a cognitive function that is going to require integration with many other brain regions. However, part of our ability to perform hearing and noise tasks is going to depend on the quality of the information that the brain originally receives from the environment. So the quality of that frequency encoding in the beginning. So we do know that hearing and noise perception is dependent upon higher level processes, particularly auditory working memory. We have done some work on that, and we have seen things like the relationship between musician advantages in hearing and noise perception and are mediated partially by auditory working memory. But it's also the quality of that original representation which count. And perhaps integration of multimodal information. Mostly what's being studied there is visual information. If you can look at someone's lips when they're speaking in a noisy environment, it becomes much easier to understand them. In this talk, though, I'm going to ignore higher level processes and just go for the quality of early sound representation and integration of multimodal information. So to get at some of these problems in healthy intact humans, we use a small signal called the frequency following response. It's essentially an evoked auditory response that you can pick up with electroencephalography, and it tells us something about the fast temporal dynamics of sound processing. It's sort of equivalent to pitch information, but not entirely, so if you feel strongly about that, don't get too upset, but it does encode frequency information that's related to pitch. So how you do it, you have someone listen to usually a repeated speech syllable, like the syllable da, which happens to elicit a very nice, strong FFR. And the vowel portion of that, the is made up of periodic information, and there are neurons in the system which will phase lock their firing patterns to each of those waves in the sound that's coming through the air. So you have to do this a lot of times because it's a very small signal. You average over about 2,000 repetitions, and you get something that looks like what I have on the left here. There are these onset transients which are equivalent to a click-derived auditory brainstem response. For now, we'll just focus on the response to the vowel, which we call the frequency following response. And I will show you some graphs later that are looking in the frequency domain. To give you a heads up on that, the fundamental or lowest frequency in the signal, we can just quantify the strength of that representation by the amplitude here. So these are just 5 individuals. This one has a very strong representation. This one has a weak representation of pitch information. We have used magnetoencephalography to try to separate out some of these sources. And this becomes interesting for some research questions because you can see where differences between individuals arise and how they are modulated by things like state. So this is showing that, at least in the cortex, the strongest representation comes from the right auditory cortex. And we can go in there and suck out those signals and look in the frequency domain, and we see that there's a nice peak at the fundamental frequency, which for this particular stimulus was 98 hertz. You don't see it in control regions, and you do see it at those nuclei along the way, cochlear nucleus, inferior colliculus, and thalamus. This allowed us to see some of the interesting properties of the frequency following response that are particular to the auditory cortex. And one of them is we seem to have some kind of oscillatory entrainment effect. And one of the ways we can see this is that there's a kind of after effect. The signal continues a few cycles longer than the length of the actual stimulus. This paper here goes into that in a lot more detail and why we think that's a real mechanism of entrainment. And this paper here is a kind of a group analysis with a number of different labs who are looking at oscillatory entrainment throughout other perceptual systems and trying to determine if it's real or not. In our opinion, for now, which is unsubstantiated because we don't have a connection to behavior yet, the purpose of this stability is some kind of stability mechanism for increasing the stability of the frequency representation. So one can imagine how this would be useful in hearing a noise. If you are able to follow and track pitch information, even when other stuff is going on and covering it up or the pitch information is degraded, then you'll have a better time understanding speech and noise. Another thing is that FFR strength seems to be state dependent, but really only at cortical levels. So Thomas Hardman and Nathan Weitz published in 2019 that you can get a modulation, but only in the auditory cortex. That has to do with whether or not you paid attention to a visual stimulus or not. We have done a similar thing, but in sleep, so no attention. And we find that as you fall asleep, listening to da, 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 of course, which is not very easy, as you go into deeper sleep stages, the strength of the fundamental in the FFR decreases, but only at the cortical level. Brainstem and thalamus are operating normally. Here's just one of them showing from the thalamus. We don't see any difference as you go into deeper sleep. We also see that sound gets through. This was a thing in sleep research where they thought that maybe sleep spindles are something that blocks sound getting to the cortex. We've also shown that that's not the case. Sound definitely is getting up to the cortex during sleep. And finally, we can look at the strength of that frequency representation at different locations in the system and see how it relates to complex perception, like hearing and noise perception. And we find relationships throughout the system, but the strongest relationship is in the right auditory cortex. This is showing us that there is some behavioral relevance of this signal and of differences between individuals and how well they're representing frequency information. So far, we learned a few things that the FFR is both pre-attentive, it works anyway, even if you're sleeping, and it's affected by brain state and attention. We think it might play a role in stabilizing pitch representations. It seems to support higher level skills like hearing and noise, and it seems to be malleable by neuroplasticity. This last statement is a bit controversial. Some recent papers where people have said, no, it's just genetic differences or something that's happening beforehand. It's not really about musical training. However, there are coming out now some causal studies where people have done some kind of musical intervention, looked at the frequency following response before and after, and seen that the representation actually improves. One such study is by Low et al., which just came out a few weeks ago, and they're looking at a choir intervention. So they take older adults with a little bit of peripheral hearing damage that's uncorrected, and they split them randomly into 3 groups, a choir singing group, a do-nothing group, and a music listening group. And they find that only the choir singing group that has that act of producing and then listening and getting feedback, so there's really an auditory motor loop going on there, that's shown in blue. Those are the only ones in which you see an increase in the frequency following response quality. This is quite a small study. It's looking at, I think, 12 to 14 subjects per condition. So it's now being replicated as part of a multi-site, pre-registered, randomized controlled trial to see if this is really the case and it's replicable. All right, so that's telling us that the FFR is a bit malleable. It's potentially a target for intervention. But there's another thing we could do to potentially support people's hearing and noise skills. And from this, we take some inspiration from other species. So an alternative strategy is to think about how we could add complementary periodic information. Other organisms do have ways of capturing vibrations that are relevant for them. So if you look carefully at your goldfish, you will see that it has a series of little dots along its side. What those dots actually are holes, and they are holes through which vibrations in the water can actually pass into this tube called the whole thing's called a lateral line. And inside of that tube, they have some little sensors that are actually very similar to our hair cells. And this is another means that nature has figured out how you can get vibrations and turn them into nervous impulse that the brain can use for various things. And if you look at the neural response, it actually looks very similar to a frequency following response, because you do have neurons that phase lock their firing patterns to upstates in that vibration that's going through the water. Now, in our case, we don't have a lateral line, but we do have some other sensors that do vibrations, and they're located in the skin. The main one that's probably bracketing our frequency range of interest, similar to the FFR, so about 80 to 450 hertz, are Pacinian corpuscles. So they respond to vibrations in the environment, and that could either be passive, because you're touching something that's vibrating, or it can be active, so you're creating a vibration by moving your skin over something. And that's how we feel texture, of course. So my colleague Andrienne Sharp at the University of Laval has been experimenting with how we can augment people's hearing by giving them vibrotactile information. The way she does that is glue little speakers onto a glove, and so you feel the vibrations, and she can separate out different frequencies or different bits of information and send them into different fingers. And what she and her student, Luna and Chauvette, have found is that this vibrotactile information can enhance perceptual and behavioral performance. They do basic discrimination tasks to try to map out the functions, and they are also looking at hearing and noise like us. And it seems to help particularly with temporal cues, but they're still trying to map out exactly how you can best send the information to the system. So we would like to know, is it really the fine frequency information that's relevant here, or is it more just big amplitude modulations that maybe have to do with syllable rate, so the lower frequency ones? So we asked the question, with this glove, can we create vibrotactile frequency following responses in humans? So we took 41 young adults, actually we didn't, Anna did, who is here today somewhere, and we recorded 64 channel EEG in 3 conditions in the same young adult subjects. In the first one, they got only auditory stimulation. The second, they got vibrotactile stimulation and some auditory masking, so they wouldn't accidentally hear the little buzzing sound that might be produced by these speakers on their fingers. And in the third condition, they received both at the same time. So first, we just made sure we got an auditory FFR with a setup. Looks good. That's what it looks like, nice and strong. We've got a fundamental response here, and we have integer multiples, which are the harmonics. And then we went to look at the vibrotactile by itself. So this means they don't have any auditory information. And we in fact see that there is a frequency following response. And it's not as strong as the auditory one, but it's very clearly there in all of our subjects. Another interesting note here is that it doesn't have any harmonics. It's only got the fundamental. So there's a difference in the information that's being represented. It's not really shown here, but it actually does have that after effect that I mentioned earlier is characteristic of the auditory cortical FFR. We can look on the topographies as well to see how these things relate. This is what a typical FFR looks like, a frontocentral distribution. And if we look at the equivalent for the vibrotactile stimulus, we get this. We presented this on only one hand, and it was the left hand, so we're getting a hotspot on the left side. We're guessing that's going to be primary somatosensory cortex, but we haven't done anything with source localization yet, so that's just a guess. So our question is really about how is this information being used and integrated. So we were wondering, do the auditory and tactile FFRs reinforce or relate to one another somehow? So the first thing we did was just a straight correlation. If you have a strong FFR, auditory FFR, do you also have a strong tactile FFR? And the answer is no. There's no relationship with those two things. These are quite separate systems. Second, we went looking for super additivity. This is the idea that the combination, having both inputs simultaneously, auditory and vibrotactile, would be giving a stronger FFR than the auditory by itself plus the tactile by itself. That is not true at all. We don't find any evidence of that in any of the channels. So there's really no super additivity. So what we think this means is that the two signals are kept quite separate, at least at this level of representation, and integration takes place at some other later stage, which is not represented anymore in the frequency following response. So our colleague Philippe Albouy at Université Laval, sorry, that was half in French, half in English, is following this up with some intracranial recordings to make sure that we can really see a tactile FFR. And it turns out that we basically have confirmatory results. And so he took 10 patients with epilepsy and implanted intracranial electrodes. Well, he didn't do it for the study, of course. And he finds, as usual, an auditory FFR, which is concentrated in the right auditory cortex. And in the tactile modality, this is tactile only, he also sees a slightly smaller but still very present tactile FFR. Also found no evidence of super additivity, and this one seems to be a bit more distributed. We don't have very good coverage yet to see what's going on there. This will be presented in a poster on Thursday, number 294, by Robert Sartori. Unfortunately, Philippe couldn't join us today. So in summary, the quality of encoding affects real skills in humans, like hearing and noise perception. These can be modulated by top-down processes and probably by neuroplasticity. And tactile FFRs exist, and it seems like tactile information can be quite useful, but these two systems, at least at the level that we observe with FFR, are separate and in different brain regions as well. Sensory integration is behaviorally relevant, but we don't see it with the FFR, so it's at a later stage. And that's about it. Thank you for your attention.
Yeah, thanks again, Emily. So, are there questions?
Hi, Mark.
Hi, Emily. Quick question is, you mentioned there are no signs of super additivity, but is it sub-additive? Because there can also be a sign of integration, of course. It might be harder to prove because of the volume conduction EEG.
At least on average, there's no difference in any direction, but it's possible there might be subgroups or individuals might be doing different things, because we do have quite a bit of distribution, if you see on the right plot. On average, there's no difference in either direction. That's a good point, though. Thanks.
Thank you. That was very nice. I was actually going to ask close to the same question, but a second one too. Did you do any manipulation of the temporal relationship to account for differences in processing time? To maybe so that they might overlap?
What do you mean? Presenting the stimuli at
Different SOAs?
Yeah, no, not really, not yet. This is the very first study on this. So we just needed to get as good signal-to-noise ratio as possible. So we just packed them in as closely as possible. But that would be quite interesting.
Do you expect maybe that might show some super additivity?
Possibly. There are definitely predictive processes in there, also operating at the higher levels. So one could imagine that if the tone, or not, it's not a tone, it's a syllable. If the syllable coming in is being predicted versus is unpredictable, you might have a different degree of integration. It's a very good question, but I have no idea what would happen in that case. Thank you.
Hi, thanks. That was a great talk. To what extent do you think that the cortical and the subcortical FFRs might differ in how important acoustic versus perceptual information is. So for instance, if you had tones with a missing fundamental.
So the missing fundamental I've only measured at the scalp using EEG, and you definitely we have a paper about that 2017 and +1. But anyway, it's you do see the missing fundamental coming back in, and it differs by person. But I don't know where that's coming from because we didn't have full coverage. We only had a few electrodes for that study.
Are there other questions?
This one here.
Oh, yeah, there's one.
Hi, thanks. Do you also see an FFR if you use less temporally regular stimuli? And if so, do they show this oscillatory pattern that continues after the offset?
The short answer is we haven't looked at it. The long answer is I'm quite sure that you would still see it. So not if it's skipped altogether. The FFR is always driven by something. You have to have input coming in, otherwise it doesn't do anything. But I suspect that if you present it with any degree of regularity, you're still going to see the FFR and you're going to see the after effect. That's been my impression from what I've been playing with so far. It could be the case that the degree of predictability of the stimulus's appearance overall will somehow change the amplitude or maybe even the duration of the after effect, but we haven't looked at it yet. Thank you.
So we would have time for more questions. Doesn't seem to be the case, so let's thank Emily again.
Thank you.
And we move on.